We acknowledge that we are on Treaty 6 territory, the gathering grounds of many diverse First Nation, Métis, and Inuit peoples, whose footsteps have marked this land and whose presence continues to enrich our vibrant community.
Hello, and welcome back to Research Recasted, the Knowledge Mobilization podcast. I'm your host, Kelsey Howlett, and today we are joined by Dr. Trevor Hamilton. Professor Hamilton is a neuroscientist who obtained his PhD from the University of Alberta, where he studied electrophysiology and electrical potentials of individual neurons. After beginning his faculty position at MacEwan in 2009, he started a behavioral neuroscience laboratory for zebrafish, which he continues to this day. Trevor currently holds a Chancellor's Research Chair position and has also recently earned the Dr. Sherrill Brown Distinguished Research Award for his work with zebrafish. Hello, Trevor. Thank you so much for joining us today.
I'm very happy to be here.
Oh, we're excited to have you here. I'm excited to get into your research. And that's where I wanted us to start. Could you start by just explaining to us a little bit about what your research is currently?
Certainly can. Started at MacEwan in 2009. And back then we started a zebrafish lab for the purpose of doing research in behavioral neuroscience. And it's come a long way since then. Yeah. So we have had a lot of success in a lot of different ways, but it's kind of three main areas that kind of almost spheres of research. First one is learning and memory in fish. So we started off investigating novel object recognition testing in zebrafish that hadn't been done before.
Okay.
So we provided a nice paradigm for testing object recognition memory in fish. And believe it or not, it's longer than 30 seconds or 3 seconds or whatever the general public seems to think it is. Fish have a variety of different types of memory and memory spans that are a lot longer than 30 seconds. So, we developed this task and we've used it since then to test certain drugs that can enhance memory. So nicotine, for example, we showed that nicotine can enhance object recognition memory. We've used it in collaboration with researchers at the U of A who made these knockout fish by altering prion proteins as a potential model of Alzheimer's disease. So imagine a fish with Alzheimer's disease. If someone develops this, how do they test it? Well, we used our test to demonstrate that these fish had an age-related memory decline, much like you'd see in Alzheimer's compared to controls.
Oh, interesting.
Yeah, kind of random, kind of fun, and beneficial because zebrafish are a really important animal model in research. So that's kind of sphere one, memory with object recognition as well as episodic-like memory. And another sphere of research would be ecotoxicology. So we study the effect of different pollutants and toxins on the behavior and physiology of zebrafish. So in the past, we've studied oil sands, processed water. We studied benzoate-pyrene, which is a component of crude oil. We've studied 6-BPD quinone is a recent one. So that's a component of tire wear runoff. So, you drive around your car and where do your tires go? Because obviously they go somewhere. Mm-hmm. They wear down onto roads and asphalt. And a component of that, if there's a big rainstorm, can rush off into rivers. And it's thought that it may have caused a big salmon die-off a couple of years ago. So it became a topic of interest. And so we've studied that in the lab as well. We've also looked at the impact of elevating carbon dioxide causing aquatic acidification and how that impacts fish behavior. So that's kind of the ecotoxicology sphere. And the last one is psychopharmacology. And so we've investigated the impact of acute or repeated drug administration on fish behavior and physiology. So we have, drugs like alcohol and nicotine and terpenes that are really relevant now because cannabis is legal. So we see a lot of these terpenes being consumed by people, and there isn't much knowledge about what they actually do. So we've studied some terpenes. We've studied some drugs that increase anxiety, so anxiogenic substances. We've looked at repeated and acute exposure in models of addiction. So that's kind of a little spiel about the kind of the 3 areas of work that we do in the lab, and what we've done over the last 15 years or so.
Why zebrafish? You said that they were like an important species when it comes to stuff like this. Why is that?
It's a really good question. First of all, the fact that they're the 3rd most popular model species for medical research. So rats and mice are number 2, but number 3 is zebrafish. So it's commonly used in the medical world to study everything from, stroke and cancer and heart disease, as well as a lot of neurological issues. So it's a popular model organism. And with that, there have been a lot of really neat scientific tools developed to study zebrafish physiology, brain chemistry, neurochemistry, all of those sorts of things. For us, they're a lot cheaper than rodents, so we can house them at MacEwan. We don't need, a massive basement facility to have rats and mice, so it's convenient that way. They also reproduce really quickly, so we can have fish after about 3 or 4 months make another generation, so we can start looking at transgenerational effects of toxins or drugs. So yeah, there's a lot of really good reasons why they're important medical models.
So they're medical models, so that means that they would be really similar to us as humans.
To a certain extent.
Yeah.
So a lot of the neurochemical pathways are conserved. So, the neurons themselves in the brain of a fish or a rat or a human all have the basic fundamental properties of firing action potentials and EPSPs and all the fun things that my students love to learn about in 275 and 375. And the same neurotransmitters are also present. So it's probably a good time to ask you if you'd like a little show and tell on the show.
Honestly, that would be great.
Okay, perfect. Because I have a young son in grade 1 and he loves show and tell. And I thought this would be a great opportunity to do a similar thing and show you some brains.
Oh my gosh, amazing.
Okay, so here we go. So this is specimens of 2 zebrafish brains along with their eyeballs attached. So this is an example of a fish from our lab that were dissected by a student of mine, Ethan. Did a great job. And in those brains, there's about 100,000 neurons. And so
oh wow.
The next one that we have are some rat brains. So these brains are specimens from our collection in the psychology department. A rat brain has about 200 million neurons, so a lot more. And the third specimen we brought is a human brain that's real, but only half of it. So we have half a human brain.
Wow.
So this, in the human brain, we have about 86 billion neurons. So an insanely high number of neurons. 86 billion is hard to even imagine.
Yeah.
So we have, half of that. So there's 43 billion in this specimen here. So yeah, any questions about the brains?
Honestly,
you want to check them out? So those zebrafish brains weigh about 5 milligrams, so hardly anything at all. A rat brain is about 2 grams, and then the average human brain is about 1.4 kilograms, about 4 pounds.
Yeah. And you've only obviously have half of it here.
Yeah.
I was very curious. So we've all seen images of the human brain, photos, you see it in TV, movies and stuff. It's quite different to actually see one in person. But I was so curious as to what a fish brain would look like. And that's not at all what I was expecting. Kind of little white and I guess you said that the optic nerve and the eyeballs are still attached.
Yeah, it's kind of nice to have the eyeballs there because it gives you a bit of perspective and you can actually see the brain tissue itself. Without those eyeballs, it would be really difficult to see. So it's nice that they're present. And those are adult zebrafish brains. The larval brains are obviously a lot smaller. So when we perform certain experiments that are, with repeated drug exposure or toxin exposure, often we'll dissect the brains and we'll collect them and we'll analyze the neurochemistry of the brains themselves to see if there are differences caused by whatever substances we applied.
Yeah, okay. So when you're studying this addiction and stuff in the brains, obviously you have to get them addicted to something. How does the process work for that?
Good question. Because we can't obviously offer the opportunity for the fish to take substances. It started a long time ago. One of the first papers we published was, we looked at the impact of daily moderate alcohol versus weekly binge alcohol administration in zebrafish. It was a topic that one of my undergrad students was interested in, so I thought it's a good research question. And so we gave fish, the way we do it is we place fish in their housing tank in a separate, it's called a spawning tank. So they can be together in a tank. And then daily, we would put the desired amount of alcohol, in this case, in another dosing tank. And then basically take the container full of all the fish from their habitat tank into the dosing tank, and then administer alcohol for the desired amount of time. So when we did it repeatedly for, it was about half an hour a day, we found that after 21 days, we stopped and gave them a period of drug withdrawal. We found that the fish were behaving obviously differently. So the drug had an effect. And they had a preference for the side of the tank where they were, or the stimuli around the tank they were originally dosed in. So they're dosed around white walls, and we found all of a sudden these fish really wanted to go towards white, and that effect wore off 9 days later. So that, led us to start thinking about whether or not these fish were actually desiring the situation where they were given the alcohol, which in psychology we know is conditioned place preference. So we've kind of continued with that model in a lot of different ways. But just to go back there, the binge dosed fish, their behavior was very erratic and very messed up for a long period of time. So that told us initially that, not that you should definitely do one or the other, but binge drinking is worse than daily moderate drinking, especially if you're a fish. So now we do a similar type model where we give them alcohol or nicotine for an hour a day, 21 days, and then we look at their drug withdrawal. So we've shown that if we place a red stimuli around the dosing tank and then test them during drug withdrawal, they'll prefer that red color over another color. And we've counterbalanced everything. So that indicates that these fish seek out the situation that they were given the drugs in, which is drug-seeking behavior, which is a component of addiction. And the second one would be testing for anxiety-like behavior. So we can look at whether or not a fish is essentially stressed out and demonstrating what we would say is anxiety. But we can't ask the fish, are you anxious today? Because it can't obviously communicate that. So we have to use these metrics to assess anxiety, and we call them tests of anxiety-like behavior. So after drug administration and withdrawal, if we give them one of these tests, the similar effect happens, and we see that their anxiety is increased, much like you'd see in a human who's addicted to alcohol or nicotine. And so after a while, this does wear off, just like it does in humans. But the whole idea is developing a paradigm to test addiction of a variety of different substances, and then also use other pharmacology or other measures to potentially treat the anxiety and the drug seeking during withdrawal. Make sense?
Yeah.
Okay.
No, it's just, it's so fascinating thinking about how fish experience the same behaviors that we do, because I would have never have equated the two. So I think it's really fascinating that you can do all this stuff with fish. I am curious, what is, and this might just be for my own interest, what is the lifespan of a zebrafish? So how long do you have them for? Because I know you said that they repopulate very easily.
Yeah.
So I'm just curious, how long do they normally live?
In the wild, less, because they're predators that can pick off the slow older ones. But 3 to 5 years would be the maximum life expectancy of the fish. And ours, we usually end up testing the adults between the ages of usually 5 and 12 months. But at the same time, we're finding that at those different life stages, there are differences in their baseline behaviors, as you'd kind of expect, because, if you think about yourself at 25 as an adult versus 45 versus 65 as an adult, your behavior is going to be a little bit different. You are different. And so we're seeing a similar thing in zebrafish, and we're actually doing some experiments related to that now, which are being conducted by some great independent study students.
Huh. That's so cool. Speaking of independent study students, you currently have a Chancellor's Research Chair position here at MacEwan. And I want to ask about how that is impacting your research and what opportunities has that provided with you to expand on this?
A lot. It's been fantastic. And so with that award, it's given me the opportunity to teach in a little bit of a different way. So I don't have in-person classes, but instead I've taken on more independent study students, and I'm working more with my honor students and research assistants. And so I have at the moment 5 independent study students, and I've never had that amount before. It's a lot of research projects on the go, but it's been fantastic. So I've got Siddharth is working on a really interesting group intruder paradigm where we're looking to see if you've got a group of fish and you introduce a stranger, if that impacts the way that those fish behave. Again, a kind of a model of increased stress in fish. And so we're working on, manipulating the ages of those shoals to see if this is a realistic way of testing group intruder stress. So that's one of the projects that's going on. It's going well. It's lots of fun. We've got another project that Madison and Gary are working on where we're going to give different ages of fish nicotine at different doses to see if there are differential effects of nicotine across these different ages.
Okay.
Something that's never been done before, so it's fun and exciting. And then Navi and Alex are working with a new system we have to study larval fish behavior. So larval fish are super small at 5 days old after basically they hatch. And we've got a system where we can study up to 24 at the exact same time. So we put them in these wells, and it allows us to test novel compounds or drugs in general to see what the impact on behavior is. And so for their projects, I had an email out of the blue from a person at the Marie Curie Institute in Poland, and she makes drugs, makes novel compounds. And she emailed me and said, your research is very interesting. Would you like to collaborate? And I can send you these novel compounds and you could test them on your fish. So we used more scientific terms, but basically that's the gist of it. And I said, sure, that's great. And so Alex and Navi are testing these potential opioid-like drugs to see the impact on the fish behavior.
Wow.
So there's lots on the go. And I would normally not be able to take on this many projects, but with a little bit extra time, it's been possible, which is great.
Yeah, amazing to have so many projects and to also be able to allow the students to have the opportunity to partake in the research, which is so fantastic because a lot of students, especially at the undergrad level, don't really get opportunities like that. So it's really cool that you're able to actually have these students in the lab helping you out. I also want to ask, and first of all, congratulate you because you've recently been awarded the Dr. Sherrill Brown Distinguished Research Award. And I want to ask what kind of impact that has had on everything going on. Yeah,
thank you. That's another award I'm very happy to get and very proud of. And that has been, it's great in terms of having the prestige that comes along with the award. So it's really important when you apply for external funding or even internal funding to be able to say, not only have you done research, but your institute has recognized you for your achievements and for how well you've been doing. So that was great. There's some funding involved as well. So that's going to allow myself and a couple of my students to travel to Toronto for a Canadian Association for Neuroscience conference. So we'll get to travel there and present some research and meet other researchers and collaborate, which is good. It's been, COVID was not ideal for a lot of things, but especially science and connecting with other researchers. And, there were some students in the lab at the time, and they just didn't get those same opportunities, which is too bad, because going to a conference, and, we have a great MacEwan Research Day as well. So I encourage everyone to go to that. Just meeting other scientists, talking about like-minded things. You can walk up to a poster and you can say, you can know nothing about a topic and say, can you please walk me through this? And then ask all the questions you want. So it's a great opportunity to just learn and, learn about things that normally, in class, you may be afraid to ask that question, but at a poster session, you can just have a casual chat and learn some new things. So conferences are a fantastic way for scientists, especially young scientists, to learn material and get excited about the scientific method, to get excited about zebrafish or rodents or humans or whatever it may be, and, progress their careers.
Yeah, wow. No, that sounds like an amazing opportunity for you, for the students, for everyone. Where do you hope this research chair position, this award, where do you hope it'll take you from here, in the future?
I hope, to continue along the path we're on. The research chair has also given me time to focus a little bit more with some collaborators I have in the States at the University of California, San Diego, at the Scripps Institution of Oceanography. And we were recently awarded a pretty sizable grant to study the neurobiology of changing oceans. Which it's a very broad but very important topic. So it's this incredible dream team of researchers put together by a collaborator of mine. I feel like I'm the random Canadian on the team, and I'm glad I'm there. I've done some work in the field, but everyone else, they're researchers who work with corals, snails, sea urchins, and stickleback fish. And the idea is to study how hypoxia, so lowered oxygen levels, increased temperatures, and increased CO2 causing ocean acidification, how that could potentially impact all of these different candidate species. And so we want to look at creatures in the wild, as well as bring some back to the lab to then manipulate some of these experimental variables and look at, the winners and the losers and the resilience of some of them and not others, and kind of get a really good understanding of what is happening at a neuronal level and see what's happening to essentially the brains and the nervous systems due to these challenges that are probably going to happen in the future. Yeah. So I'm excited to have a bit of time to focus more on that project as well.
Yeah, that's also a very exciting opportunity. Changing oceans. So is there a specific ocean that you guys are focusing on, or are you guys going to kind of...
The Pacific is where we'll collect most of the species. So we're going to look at stickleback fish all the way from Alaska down to San Diego and collect different populations to study. There's a population geneticist who studies stickleback, something that I don't know anything about, but, analyzing how the genetic variability is could be similar, and we'll pick some of those to bring back to San Diego to then look at multiple generations of those fish and use similar techniques to what we use in our lab at MacEwan. So we'll probably study the larval behavior, we'll study some pharmacology, and we'll study a whole pile of things in these stickleback fish.
Yeah, wow. So I'm just making an assumption here, but different fish are very different in how they operate and they grow and they their brains work. So it's not kind of a one size fits all sort of thing.
Yeah, no. So in terms of fish in general, there's over 30,000 different species of fish on the planet. So there's a huge diversity across species. We're going to pick stickleback, but we're just going to look at different populations along the coast. And if you think of, a fish that lives in Alaska and the temperatures that it deals with in the ocean compared to San Diego. It's if you have to pick a place to go on the beach and maybe surf or do whatever, you'd be down in San Diego, not up in where it's freezing cold all the time.
Yeah.
So it could be that different populations, because of where they're located, have more or less resilience when it comes to these changes. So we want to investigate that.
Okay, interesting. Now, So you're doing all of this stuff, partnering with different institutions like San Diego, Poland, all of these different places, and you've got all of these awards and these chair positions. I'm curious, what kind of inspired all of this for you to begin with? How did you decide, or like what made you decide that you wanted to spend your life basically researching zebrafish?
Mm-hmm. It's a good question, and sometimes I ask myself the same question. But my PhD was in neuroscience, studying cellular properties of rat hippocampus. So I looked at a lot of those brains as a PhD student. And I studied action potentials and dendritic potentials, and I did calcium imaging and these really cool techniques. But I really got interested when I got hired at MacEwan about, the behavior of whole organisms. And zebrafish are ideal because, again, we can study toxicology, so we can study how toxins or climate change could potentially impact fish in the wild. And, we're in trouble if all the fish on the planet die. It'd be a very dire circumstance. But zebrafish also are this medical model, so we can use them to try to relate to humans. And so it was just this ideal organism to start studying with a diversity of research questions. And so I thought, this would be great to start off on. And then things just kind of spiral in certain ways. And you meet people that have exciting ideas and you work with them. And so I don't think I necessarily chose the path to where I got to now. I just took advantage of certain opportunities that were there and learned a lot of cool techniques and learned a lot of ways of thinking about science and fish that 15 years ago, I never thought I would have. Mm-hmm. So in the future, I'd like to still, go back and look more at the neuronal level at what's happening in the brains of fish. That requires some very expensive equipment. So we'll see. It could be with collaborations. It could be big grants. Who knows? So there's a lot of great opportunities on the horizon. But what kind of keeps me going is how excited students are about doing research. And, MacEwan has fantastic students, and the opportunities that they have here are great. So it's great at MacEwan that a lot of students have the opportunity to actually perform real scientific research. And, I don't expect all of my students to become zebrafish researchers. In fact, I don't think any have. Not yet. Yeah. Maybe someday. But it helps them with the next stage of their career. So I really like, teaching students about scientific method and how you properly analyze science and how you think about not just the science itself, but the politics that surround science and publishing. And, there's a lot of aspects to it that unless you're in it, you're not aware of. Mm-hmm. And then students can bring this to the next stage of their careers, if it's medicine or dentistry or law or, psychology, whatever it may be. I think it's really important to have a good understanding of science in general.
Yeah, no, and I truly believe what you said about MacEwan being great for what it does for its students. I also have a degree from the U of A. And while I do not study science at all, I was an English student. It was kind of the same way. Like, it was all very academic, and there wasn't a whole ton of opportunities. But coming to MacEwan, you all of a sudden have these opportunities to work in the field, to get this on-hand experience, which I think is amazing. And I think it's so cool that you're able to help students do that, especially in the lab. I do have one last question for you before we end today. And it's, what advice would you give to our listeners, whether it be staff or students or anyone else in the world? Like, what advice would you give them?
In terms of scientific advice?
It doesn't have to be like, whatever. It doesn't have to be specifically related to your research or anything. Just kind of like, what advice would you give to somebody?
I think it's important to be inspired and find something that inspires you to be creative and to enjoy what you're doing. So in terms of your eventual career, it's probably not going to be something that's 100% fun all the time. A job is something that is a job that could be, it could be onerous. You may not look forward to it, but, it's great if you can look for a career that is at least partially enjoyable that you'd like to do for a long period of time that can keep you maintained, keep you inspired, and help other people. And I think that, being inspired and creative is key. And then also having fun and enjoying yourself in your life in general. So it's, your work is one thing, but sometimes people need to take a break. And it's difficult sometimes as an academic to separate yourself from work and life. But I think it's really important. We need to, with social media and phones everywhere, there's all this, we're bombarded with all this information all the time. And it's important to take a break from a lot of the technology and go outside, enjoy nature, do what you need to do, get exercise, have fun with friends and socialize. So, reconnect with yourself so that then that can help you become stronger when you're, at work or doing whatever tasks you need to do during the day-to-day.
No, that's great advice. Thank you so much for being here, telling us about your research. Thank you for the lovely show and tell.
I hope you enjoy the show and tell.
That was very exciting.
My son Parker would be proud. He's in grade 1 and he does a lot of show and tell. So,
yeah, has he ever brought brains for show and tell?
Not yet.
Not yet. Yet being the key word. All right. Well, thank you so much.
Yeah, thank you for having me.
Well, that's all we have for today's episode of Research Recasted. If you want to support this podcast, you can visit Research Recasted on your favorite podcast platforms to find new episodes every 2 weeks. Also, don't forget to check us out on Instagram, where you can give us a like, leave us a follow, or send us any follow-up questions you have from today's episode. This has been Research Recasted, the Knowledge Mobilization podcast, brought to you by the Office of Research Services and the Faculty of Fine Arts and Communication at MacEwan University.